Okay, we'll get started. So welcome everybody to this fourth field day of the Animal Genomes to Phenomes Phenomes initiative. If you want to know more information about this program, go to the website ag2pi.org. There we also have recording of previous field days. So today we're going to talk about dairy cattle. Before I introduce the topic and the speakers, I will also introduce two important people that are working behind the scenes. Nicole Scott, who is a program coordinator for AG2PI, and Eddie Cheng Ting, who is a bioinformatics analyst and software developer in Pat Schnabel's lab. So welcome everybody. Apologies for the change in the date, postponing it by a week, but we felt that that was an important accommodation to make. So today we're going to talk about dairy cattle, and I always view dairy cattle as sort of the poster child for genomic selection. That's where genomic selection was initially implemented in, or the first species on a large scale to implement genomic selection. And there's also a lot happening in terms of phenotyping and some unique aspects of the phenotyping that has happened historically and that is happening in the future and as we speak. And so for that we have three excellent speakers who have a lot of expertise in the area. I'll introduce the first speaker in a second after I've mentioned that if you have questions as we go along, put them in the chat, and then we'll, if it's applicable to the topic at the moment, then I'll interrupt the speaker and pose the question right there, and otherwise we'll go through them at the end. So the first speaker is Dr. George Wiggins. George is a research geneticist at the Council for Dairy Cattle Breeding in the US. The Council for Dairy Cattle Breeding is a non-profit organization that is in charge of calculating and distributing genetic evaluations and genomic predictions for US dairy cattle for the industry, and also to manage the data and distribution of the data. And it's a fairly new organization in 2015, because at that point, this organization took that responsibility over from USDA ARS. And prior to that time, George was working for USDA ARS on exactly this topic, and he was very much in charge of implementing the first animal model blood evaluations and also genomic prediction. So with that, I'll turn it over to George. So George, if you want to start sharing your screen.
Okay.
And I should mention that George also has a couple of poll questions in his presentation, so be ready to answer the poll as those come up.
Getting a little preview of the presentation here.
Mm-hmm. Yeah.
Okay, so as Jack explained, I'm currently with the Council on Dairy Cattle Breeding, and I'm pleased to have this chance to talk about our program. So just a kind of the background of what's going on here. So dairy cattle breeding has really been an excellent application of genomic evaluation, because bulls have thousands of progeny through artificial insemination, and even cows through embryo transfer. We can get many offspring from the very best cows. We have a long history of data collection going back to 1905, and it's an international program with bull semen being marketed internationally. And we have sophisticated evaluation systems for identifying the genetically superior animals. So now, just to be sure we have a little audience participation, we're going to ask you a question with some multiple choice answers. So given that we have over 9 million milking cows in the US, how many new animals do you think we're genotyping each year? Okay, well, I think we've got a pretty good idea that people agree that this probably is a fairly popular program. So it's, as they will show in the next slide, we did about close to 1 million animals last year in 2020. So the answer C of 1 million was the correct one. It's a good program, but we're not actually up to 5 million a year yet. Okay, so another aspect, in addition to the participation of the number of animals, it's also what's the uptake been. So the fact that 73% of the breedings were to bulls that the only information we had on them was from their genotypes. They had no producing daughters in which to base their evaluation. It's another indication of the uptake. Another thing we get from the genotypes is being able to monitor possible inbreeding. So we report the relationship of each animal to a sort of representative sample of the population. We update all of our evaluations every month. And again, an indication of the importance of this is that new animals have evaluations released weekly because people want to make decisions based on this information as quickly as possible. Okay, so we're drawing a contrast here. This is genomics is a great system. What did it replace? Well, we had a progeny test system, which meant that a certain group of promising young bulls would be bred to a bunch of cows, hoping to get, say, 50 progeny or so as quickly as possible, so they could see how good their bulls were. But it took 3 to 4 years from the time that bull could breed cows to when the information would be available. So the bull was 5 years old before the first evaluation was available. So this meant it was quite expensive. We had a high selection differential in that we kept only one from 8 to 10 bulls. So this resulted in an investment of over $200,000 per bull and an average bull of $350,000 to $400,000. So expensive program. So what's the history of this evaluation system? We were very fortunate that it was forward-looking to develop a repository so that 10 straws of semen from bulls that were in this progeny test program were collected and stored. Then we went on to the sequencing of the cattle genome, and then the sort of the critical issue in this was having the Illumina bead chip available in 2007. So then in 2008, we got experience developing evaluations and having research results available, enabling us to go official in the beginning of 2009 for Holstein Jersey and Brown Swiss. Another big milestone was when the lower density chip became available, called the 3K, so that extending the evaluations to females was more affordable. And then as Jack said, the responsibility was transferred from the USDA to CDCB. So where does the DNA come from? So as I said, historically, we had the luxury of having semen available. So our reference population of bulls with highly accurate evaluations could be used because we had a source of DNA for them. Blood was also an important source early on. And then as we got into cows, hair follicles, so that pulling hair from the switch was a source of DNA. But now we've moved very heavily towards collecting a ear punch as a ID is attached to the animal. We can also collect that tissue that's extracted, and that can be a source of DNA. even making a stronger connection between the identification of the animal and the actual DNA sample. Then once the genotypes get to our laboratory, we do some validation. That's been a very satisfactory aspect of working with this data, is because there's so much internal consistency you can look for. So we look at, on an individual SNP basis, we look at call rate, proportion of heterozygous, and the parent progeny conflicts, because we want to see if the SNP is performing well. And if the performance is somewhat questionable, we can ask the laboratory to investigate the clustering for that particular SNP to see if they can adjust the demarcation between the three possible homozygous, heterozygous, homozygous possibilities. We on a whole sample, if there was a problem with the entire submission, then these checks could identify a problem. We have a report card that we provide the labs and ask them to respond to any cases where the submission seems to be questionable. This chart shows our distribution by number of SNPs on the chip. As you can see, the 55,000 seems to be the most frequent now. We still have a substantial number at 9,000, but this particular chip is being replaced by a higher density one, so next year's results will be, that will be much less frequent. Now, higher density, the 777K, is primarily for research projects, and the ones around 100,000 or so are primarily for bulls that have been identified as being superior and who's going to have their semen marketed. So by going to a higher density chip, the owner can improve the accuracy of the evaluation. So bulls are typically genotyped initially at a lower density, and then if they're returned to service or marketed at a higher density. This chart really shows the change in the sex composition of our genotype population. The number of bulls has been pretty much constant across time, but the number of cows has shown a very steady increase. And you might ask, why are they doing the cows? Well, I happen to have a slide for that. So the idea is that they're buying information. So by genotyping the cows, they can improve the accuracy of their culling and breeding decisions. So with respect to breeding, they can identify which cows to apply which reproductive technology to. The cows that are the very highest are ones that they want to collect embryos for embryo transfer to use other cows as the surrogates to carry those calves. The next level down are cows that they want progeny from, so they use sex semen with those, so they experience some reduction in fertility, but they have a more of a 90% chance of getting a female. Then the other cows that are still valuable for milking, but they don't want replacements from, they can breed to beef bulls and increase the value of the calves that they don't need for breeding. Also, there's a chance to avoid inbreeding by looking at the comparison on a genotype basis rather than just based on pedigree and make more accurate culling. Our whole program has really had a strong international bent. From the very beginning, we shared all genotypes with Canada because in the beginning, in order to get adequate size reference populations, you wanted to get every genotype and every genotype of every animal that had a accurate evaluation that you could. We also collaborated with Italy and the United Kingdom because they provided bulls that were heavily used there and the genotypes. The breeds other than Holstein, of course, have a challenge of having sufficient numbers to be able to have accurate results. So the Brown Swiss, from all the countries that were participating in the interbull program, pooled their genotypes and had interbull run an analysis so they could get the benefit of data all over the world, and also shared those with us. So our Brown Swiss evaluations have benefit of the large European populations. Similarly, Jersey has a significant presence in Denmark, so we've had some exchange with Denmark. And for Guernsey, again, being a numerically small breed, we had a collaboration with the United Kingdom to kick off our Guernsey evaluation, which was started in 2014. And Japan, Switzerland, and Germany have fairly routine exchanges where they get the benefit of our large reference population, and we get the benefit of having additional bulls in our system. So that's kind of on the input side of what data we had to augment our reference population. This shows the genotypes that come to us because people want to get evaluations in our system. And not surprisingly, the bulk is from US and Canada. But you can see that we also have strong participation from Western Europe. And then on the next largest green there of Southeast Asia, that's primarily Japan and China with about 50,000 each, but also Saudi Arabia with 50,000. So what do we do with these genotypes? So obviously we do genetic evaluations, but they also have other things that we can deploy them for. So the parentage validation and discovery. So with our 4.5 million genotypes, we have confirmed sires for 97% of those and confirmed dams for close to 40%. We routinely compare each new genotype with all the other genotypes we have in our system to discover parent-progeny relationships that weren't reported with a pedigree or identical genotypes. So this is partly on for quality control and also for helping fill in missing or doing parentage discovery. So when parent-progeny conflicts are detected, we report them to the breed associations and the requesters, and usually we can provide a discovered sire. As part of the quality control, animals with incorrect sire dam are excluded from evaluation. Imputation is a really critical technology in our system because in order to accommodate that wide variety of chips, while evaluating all animals with the same set of SNPs, we need to fill in the missing observations, so to speak. So that's done by taking our genotype and splitting it into the maternal or paternal contributions. It's called phasing. And then we can fill those in by associating them with ancestors and descendants to try to fill in the missing data. Part of this is breaking each chromosome down into a number of segments, which we call haplotypes. And then by seeing what existing haplotypes in our library, the haplotype of this animal matches, we can fill in. So the important thing is that enables us to use lower density chips, and that all chips can be merged by imputing the SNPs that are not present. Another source of genomic data is full sequence, and we've been fortunate to be able to participate in the 1,000 Bull Genomes Project, which has collected, has participants throughout the world. As this shows in red, these are the organizations that have contributed genotypes, and the dark green is which breeds are represented. To be more clear on exactly which breeds they are, this shows that of the close to 4,000 full sequenced animals, we have 77% as Bos taurus and 8% as Bos indicus, and the other percentages various other species. Another aspect of mining the genotype information for information is that we found that occasionally when we were looking at these haplotypes, that there would be, based on the frequencies, a dearth of homozygous cases. So we decided that when there was a significant number of homozygous animals, as expected, but unobserved, this is perhaps due to early embryonic death, that when you have those two identical haplotypes together, the animals don't survive. So with a large number of genotyped animals and affordable DNA sequencing, we'll be able to use this information to discover some additional deleterious recessives. We call it haplotyping affecting fertility because that's basically how it's observed. The cow's bred and ultimately she doesn't sustain the pregnancy, so she has to be bred again. Now, the reason I mentioned the sequencing is that because we have the sequence data available, we're able to take these locations, basically discovered an area that's suspect, and using the sequence data to, in most cases, find a cause to mutation which would explain this early embryonic death. Another aspect that arose when we were reporting that we had a parent-progeny conflict, another data would say, no, that animal is confirmed as a sire. So in looking at it more carefully, we found that all of the conflicts were concentrated in one chromosome. So that could be explained by there being a deletion in that area, or even that the animal inherited the same chromosome, both chromosomes from the same parent. So we now have a bypass to accept that it is a confirmed parent-progeny relationship. In spite of these conflicts, we have 212 cases so far of discovering this kind of a chromosomal abnormality. So with parent-progeny relationships, we can be basically certain that because we have enough markers in common that we can say that this is a parent-progeny relationship. We also can extend this to grandsires with less certainty. So for grandsires, we will say that a particular grandsire is unlikely if it has more opposite homozygotes than our threshold, which is based on saying that 99% of the true animal grandsire cases would have fewer conflicts than what we've observed. Going back to our work with the haplotypes, we actually can check to see whether a particular grandsire is likely based on a haplotype comparison, which is more accurate than just one SNP at a time. And based on that, we can suggest discovered maternal grandsires. But animals with unlikely grandsires are excluded from evaluation, again, from the idea that we want to have confirmed pedigree. So we want them to get the grandsires corrected before we release an evaluation. Okay, so I have a poll here. I'll give you a little bit of a head start here, but wanted you to guess just how many traits the producers might use in their breeding program that we would evaluate. So it looks like the majority of people got the right answer, that we have, it's 44 traits that we evaluate that to provide evaluations on. And this this slide shows how we have gradually added to our set of traits that we provide over time, starting out with the milk and fat yields and then going up to recently adding health traits, and there's been a work on reproductive traits and calving difficulty traits. So over time, we've added to the data that we use to provide information back to the producers. We recently added these traits. The early first calving is based on age at first calving, another thing related to fertility. Health traits were extended to Jersey in April of last year. And our most recent was a feed efficiency trait called feed saved, which is composed from residual feed intake and body weight, so that we can see how efficient the animal was based on the feed and have some measure of her maintenance cost. Then heifer livability was also added then. So what data is all of this based on? So the milk weights are historically have been from recording one day per month. Then it was reduced often to just a single milking each month. And that's been collected through the DHI program or Dairy Herd Information staff. The fat and protein percentages, initially just fat, but more recently protein as well, and somatic cell, were from monthly samples sent to participating labs. Inseminations, reasons for removal, calving difficulty, reported through the same data collection scheme for the milk, the production information based on monthly reporting. Confirmation or type data has been recorded by breed association appraisers. So this gives a sense of the data quantities. We're in the millions for most of our traits. The feed intake is just very recent. so that and it's very expensive to measure. So we're at the 6,577. Just to give you an idea of where this comes from, because this is based on cooperating research organizations. So you can see that we had an initial program with the National Institute of Feed and Agriculture that had an initial grant that gave us about 5,000 observations. Then more recently, the Foundation for Food and Agriculture Research has helped support the ongoing collection of this data for the total of the 6,577 that we currently have. This is an ongoing program, and the data will increase, and we're always looking for ways that we can extend this data. So the general idea is that one of the aspects of genomics is that it's easy to extend to additional traits. You just have to get enough animals with data to create a reference population. And then every animal that has a genotype, you can provide an evaluation. So a genotype's good for all traits. Traditional evaluations are required because we have to estimate the SNP effects. But we don't actually have traditional evaluations for all traits of possible interest. Heat tolerance is one that we may add eventually. A concern is that in the case of, say, the feed efficiency, well, the research populations that provide this data that are expensive to measure, but will those evaluations really be appropriate for the target populations? So it's we need data. That's the bottom line. Another area that we worked on to extend the utility or the reach of our evaluations was to crossbreds. The basic problem was that the SNP effect estimates are breed specific, so that a marker might be associated with high production for one breed and much less or lower production in a different breed. So the concept here was kind of simple that, well, let's evaluate these crossbreds for each breed that we have evaluations for, and then just weight these resulting individual breed evaluations by the breed proportion. But then the sort of neat thing here is that the breed proportion is just another trait. So we have an evaluation for an animal saying it's 20% Jersey and 80% Holstein, for instance, just as another trait, so that we use the same SNPs, the same nearly 80,000 SNPs that we use for evaluating all the production and fertility and other traits to evaluate the breed composition. So when we do this, we say that animals with that are more than 90 or more % of a particular breed, we treat them as purebreds. If those are less, we apply this weighting process. The reference population is further restricted to be above 94%, so that we're sure that our reference population is specifically representing that breed. Now, there are another aspect of doing this is that we have to have all the evaluations on the same base across breed. And that's, we have that possibility with most of our traits, but on the type traits, those are within breed. Having difficulty in health traits are not available across all breeds. So for those traits, the only an individual evaluation based on just a single breed is reported. And this slide really explains why the program has been successful. And it's because once the genomics were implemented, we had a precipitous decline in the generation interval. In other words, the age of parents at the time their progeny are born, because we were able to have accurate evaluations of potential parents as soon as a DNA sample could be taken. And then those were used very heavily to create the next generation. So generation interval is basically declined for all the paths of selection. Of course, when we have selection, we have inbreeding, and inbreeding has increased. We show two lines here, the expected inbreeding of future progeny, and that's based on creating a sample of animals that of calves that are born and saying what is the genomic relationship between this animal and that specific group. Now, the fact that it crosses at the most recent years is more probably an aspect of time rather than any change in practice, because as the most recent animals become parents and grandparents, then their contribution to the future population will increase, and it will probably just be parallel to the inbreeding of the animals themselves. So this slide goes back to the initial slide showing the uptake. This has been a very popular program. So that you see, starting out, going back to 2011, less than half the breedings were to these young bulls, where it's gone up to now the 73%. It may be plateauing there. Correspondingly, the traditionally proven bulls have become a smaller and smaller percentage of the mix of breedings to AI bulls. Now, this is a slide that we usually show to say, yes, this has been an effective program. You see, going back to 2000, we had an average gain of the overall value of $14 per year, and now we're up to about $80 per year. So a dramatic improvement. Now, you could say if the genomics started in 2009, why did it increase in 2005? Well, improved practices were used. The studs were focusing more on the traits that go into this net merit dollars that we're measuring this on. And then we have continuing improvements in our selection intensity. So we're even getting some improvement even during the era of genomic improvement. But what we really point out that just comparing the current $80 a year with the $40 a year, we've doubled the rate of genetic improvement. Now, I mentioned net merit, and you might say, well, what's that all about? Well, it's of course would be difficult to have a program that you individually selected for 40 some traits. So we try to create an economic index that weights those traits by their contribution to the overall value of the animal. So this shows kind of a breakdown of the traits and what we include. And you can see that fat production and protein production have the biggest proportions. And then the various health traits altogether have some contribution. Productive life, their PL, is also important because that's basically kind of a composite measure of all the things that would affect how long the cow can last in the herd. So this chart gives us how this index has developed over time from when it first started just being a combination of milk and fat, and then has other traits that become available, how it's increased, how the number of traits has increased. And of course, there has to be an adjustment of the weightings. The value of milk has, of course, gone down because the components are the most valuable parts. We're expecting a further adjustment this year, which will take into account the feed save trait and also more recent research on the cost of production, so that the feed costs related to production of fat and protein in particular can be more accurately considered. So in the future, increasing number of cows genotype, we expect those trends to continue. Falling cost per SNP genotype, thus, as I was pointing out, higher density SNPs will be used, and even genotypes based on sequence data will be come into use. Evaluation of more traits, because the more traits you have, the more accurately can predict economic merit. Increased use of genomics and mating programs, so we can find the best particular bull to mate for a particular cow. diversification of records, more precision tools, basically more and better data, maybe even test herds to collect data that can't really be collected as a byproduct of the production system. So why is the U.S. a leader in this system, and which we are? We're an important source of genomics throughout the world. So we have the population size that can give us a selection intensity. We've had a focus on dairy breeds. We have animals producing in a diverse set of environments, have a competitive AI industry that keeps pushing to maximize the value of their offerings, independent evaluations so that there's high industry acceptance and belief in the results. So just to summarize the benefits of genomics, so determine the genetic merit of animals at a young age. That enables you to increase the selection intensity and get an increased accuracy of selection. We've been able to reduce the generation interval, more than doubling the rate of genetic gain. We've been able to identify genetic defects and reduce their frequency. We had a parentage discovery. This actually has a feedback because in the traditional evaluation system, as more of the parents are confirmed, then we have a small proportion of animals not contributing to correct SAR in their evaluation. In herd management, calling the low end replacements earlier, and lower. We can use lower animals to breed to beef bulls. So in summary, U.S. genetics is recognized as the source around the world in many breeding programs. We have the largest genetic base and high selection intensity, produces elite bulls and cows. Genomics has revolutionized animal breeding and brought some excitement to the process. International collaboration has been important to the program's success. Thank you.
Thank you, George. Excellent presentation, lots of information. And again, if you have questions, put them in the chat. There's a couple already, and we got a couple of minutes, not too much time. One question was, how does the system control for environmental effects? And of course, the data comes from herds all over the country and actually all over the world. So how do you account for environmental effects? And then maybe I'll add genotype by environment interaction. How is that accounted for?
Well, of course, you asked difficult questions, but the answer to the first question is that I mentioned the historical nature of our evaluation system, and that's been an ever-present concern to properly evaluate animals even before the advent of genomics. So our whole genomics system has the benefit of sitting on top of an accurate evaluation system developed over many years. But the fundamental answer is that every cow is considered to be part of what we'll call a management group, and her performance is really in comparison to other cows in her management group. An early advance was to consider the genetic merit of the cows in the management group, so we didn't unfairly account that if it was a high genetics herd, that the cows in that herd would be disadvantaged because they were competing with high producing contemporaries. So it's the kind of the simultaneous consideration of the management and the genomics. So with the animal model, with the simultaneous estimation of the management and genetic effects, we believe we've accurately taken care of that. Genotype by environmental interaction, we've historically had a herd SAR effect, which was somewhat addressing that particular consideration. But in general, that's a research area that we will be looking at, as we refine evaluations, how do we basically take into consideration which animals will perform well in which environments.
Yeah, and I think that's an important topic for this whole area of AG to PI, animal genome to phenome. So we need to understand better how the environment affects performance through genetics. So another question, the net merit index, how are the weightings on the different traits decided?
That's a lot of research goes into that, and it's dependent on research of many other groups, because we have to understand what the economic value is of a unit change in a trait. And then we also have to consider the correlations among traits, because a number of traits can all affect the economic value of the cattle. So the weights are take into account the economic information of the value of a unit change in production and the genetic correlations among traits.
Okay, thank you. There's some more questions in the chat, but unfortunately we're out of time. We'll save those and we'll ask the speakers to address those later on, online or offline. So the next speaker is Dr. Miel Hostens. And before I introduce Miel, one of the things to realize that in dairy cattle, a lot of the data that is used for genetic evaluation is actually recorded for management. And George alluded to that, and that's kind of unique. You know, farmers need that data to make individual decisions on individual cows, and that's the focus of the next set of the two talks. And both our next presenters, they come at it more from a precision management perspective rather than a precision breeding perspective, but just like we have in the past in dairy cattle, the data that's recorded for management can be used for genetic evaluation. And so our first speaker is Dr. Miel Hostens. He's an assistant professor at the Faculty of Veterinary Medicine at the University of Utrecht in the Netherlands, in the Department of Population Health Sciences, as part of the Farm Animal Health Program. And his focus is on herd health management in relation to precision livestock management and big data in dairy cattle. So Miel, floor is yours.
Perfect. Thank you very much, Jack, for the nice introduction. Can you hear me all well? All right, perfect. Yeah. Then I'll try to get this going. First of all, I hope all of you see the slides. This is a little bit different than probably what you normally have, where you see the screen and the slides normally should be somewhere over here. So if you don't see that, click on my head, and normally it should spotlight for everybody. So I'm a Miel indeed. I'm a Belgian working in the Netherlands and at Utrecht University. Some of you probably will know me. Some of you don't, especially talking to mainly plant-oriented people is interesting, because then we can, of course, illustrate what's going on in our industry. I have a veterinary background. I'm extremely into cows and data. Everything which is cow and data related. I never touch a goat, a sheep. As long as it's bits and bytes from cows, I touch it. Maybe I know that this might sound obvious, but this is always my introduction, because I think we have a role in that to play, a role that as researchers we sometimes forget, and that's disseminate what we are doing and why we are doing it. And I'll start with a very, very short introduction on very simply why we drink milk. I think it's important, especially in a world which is changing from the European side. People are asking a lot of questions about dairy production. So I think it's important that we tell people also the other part of the story, because they are getting scared if we produce all that data that we are doing very nasty things. And I think it's good to illustrate to people that what we are doing is something which comes from nature. It took us about 200 million years to start walking and become a mammal. And mammal comes, of course, from the Latin word mammae. This is my daughter. She's now 1 year old. This is one of the pictures I took when she was very, she was, I think, not even 24 hours old. But I'm a data geek, and even my own daughter was a subject of that. So to give you an illustration on how powerful milk can be, I started weighing her, and also trying to get her dry matter intake. So I focused at a certain moment when she was at home on the first 2 weeks of her life. And I started weighing her very intensely, and then I also monitored her intakes. So we know that people have about 10% solids in milk, or women. She has an intake of about 600 mLs, so 0.6 liters. So in other words, she has an intake of 60 grams. To give you an idea, she grows 40 grams per day in that period. Yeah, and I always joke with people, her conversion ratio compared to other species was exactly here. Yeah, illustrating how powerful this simple nutrient is. Yeah, milk has an especially for young young people, and in a very early stage, milk is extremely nutritious. Yeah, and it's illustrated by showing the feed conversion ratio. It's maybe a bit freaky to do it from your own children, but it's interesting to understand because because I have the idea that people sometimes neglect the fact that we are mammals. Yeah. Why can we drink milk? Very simple. We know that somewhere probably 8,000 years ago, yeah, there was a gene mutation on probably one man, yeah, one guy, yeah, in the tribe, yeah, and while all the other men around the village had to hunt for food, he probably, yeah, was lactose tolerant and was able to drink milk and not get diarrhea. 8,000 years ago, somewhere, let's say in the Baltic area, that happened. And we know that probably that tribe was very successful because they didn't have to hunt and were able to drink cow milk. And because of that, we have in, let's say, Western Europe, and then also at the other side of the ocean where you are at this stage, there's a lot of people which are lactose tolerant, but that's not normal. It's a gene mutation. I talk to people about that. We are a gene mutation ourselves. Why can others drink milk? There's about 5 gene mutations in the world that that allow people to drink milk. Yeah. So and this is causing, of course, milk to still be a very powerful, let's say, nutrient in developing countries, but also in, let's say, the developed countries. Although we maybe should stop talking about that. This is exactly in my head. This is something that we know. Population growth, yeah, is estimated to go towards about 11 billion people. That which means that if we are at 7, that we are going to almost double, yeah, in let's say 100 years. And very simple. Imagine that even milk consumption, on average, would drop a little bit. Yeah, then we almost need to double our production worldwide. Yeah, so that's a given. Yeah, and that's a very hard given to for people to understand. We will need to, let's say, double our production worldwide to be able to just feed the world and nourish that nutritious food, or provide that nutritious food to people. Yeah, in my head, especially when I became a father recently, yeah, having young children die because of a lack of nutritious food, yeah, it's ridiculous in my head. Yeah. I want to show you this cow, yeah, and I show her every time, and I stopped showing, in fact, this kind of metrics to people. Yeah, this is the Cells Prale Aftershock. That's a cow in Wisconsin. She has the world record in a year when it comes to milk production. Yeah, she's producing 35,000 kilograms of milk in one lactation. My excuse for not having this in pounds. Just double it and add 10%, then you have the pounds. So I think this would be, let's say, about 80, yeah, just not 80,000 pounds of milk. Yeah, but I started expressing this in a little bit different way. Yeah, from an efficiency perspective, this cow is interesting. If all cows, yeah, would be like her today, then we only would need 4% of the animals to feed the entire world. If all cows would be like her by 2100, we need 6% of the animals to feed the world today. Just to illustrate that we don't need to even further increase, yeah, but if all animals would be like her, of course, that's an average, a distribution that will need to rise. But this is very simple maths that I did at a certain moment to feed the world. But this is nice to express to people. Only 4%. Yeah, remember that the whole environmental footprint that she has, all of that is, of course, then put in a different perspective. Her license to produce, and I come to the data, what I'm going to talk about today, is of course changing. She produces 1 truck of milk in her entire life, so in her entire year, but of course she's producing 2 trucks of manure, 3 trucks of water are needed to produce that milk. Then she has about, if you look at the equivalent, 3 trucks of carbon dioxide output via the methane production. And the other thing, she has 4 plant-based competitors, which are not milk. The good thing is the EU is now going even to ban this kind of, let's say, plant juices because they're not milk. Yeah, they're going to ban even the carbon in which they are putting milk, the juice. Yeah, so it cannot resemble any more milk because it has a strange message. I saw that very recently in some newspapers that started reporting that. There's 8 daily tweets about the animal welfare, but of course, and this is my little joke, she produces about 16 gigabytes of data. This is exaggerated, although we can debate about that. Yeah, that I see there is a little bit of light going away on my screen. But what's nice to understand is that we need to start looking at us as high-income countries. Yeah, I call high-income countries developed countries. We should have a role, yeah, in showing the rest of the world on how to effectively produce nutrients using the data. We can learn from that data. Now my light is back again. We can learn from that data, but the data is a problem in the dairy industry. Yeah, we published something in 2018 about data and the big data thing in dairy cows when it comes to welfare. When you talk about data and big data, everybody always refers to the V's. Yeah, for example, the volume of the data. In dairy cows, honestly, there's not really big volumes. The only one who has big volumes probably is our previous speaker, which are the DNA, RNA sequences that are produced. The main problem is that it's scattered horizontally in different organizations. Somebody owns this, somebody owns that. The sensor data is in the cloud. The lab results are maybe even not stored. So the problem really is about delegated data ownership. And this is something that we need to become aware of. The one who can certify the data, yeah, to be right is the one who owns it. Google doesn't own the data, you own the data. They only own the algorithm. Yeah, and this is the same with cows. I see this as an extremely big problem in the dairy industry. Why? Because these sensor providers and anybody who's producing data, they are almost like putting more concrete around those, let's say, data silos, trying to protect it more and preventing us to do the right things with it. Yeah, because it really comes about combining data, especially when we talk about those more novel phenotyping purposes. Real volumes come from genotypes. I joke with people, 4 gigabytes is normally enough to store the entire full genome of one cow. But then, of course, we have the SNPs, which are low densities. That's a different way of looking at your DNA. I won't go into detail. When the RNA sequencing will jump in, this of course becomes more difficult, but that's not the goal of today. Velocity is the other one. So we had volume. Velocity is the speed at which we are creating. This is one of the cows from Kentucky, yeah, where we had the cows with all different sensors. Now that farm is almost gone, if I'm not mistaken. But now I started at Utrecht doing also a European version of that. I have a farm with a low number of animals, but I try to collect as much phenotypic information as possible. So I invited almost any sensor provider that I know, which is established, let's say in Europe, trying to have the sensors installed and start collecting phenotypes. Yeah, those phenotypes which are sometimes difficult to collect. Yeah, I'm trying to get a network also with other farms that will do the same. Yeah, and trying to create that phenotypic database for those more specific, let's say, testing that we need to do. The test herds that George was talking about. The volatility is the other problem. We're losing a lot of data. Yeah, there's estimations in science that all the data vanishes after 5 years, but that's a pity. Imagine that we would be able to reuse all the dry matter intakes that were ever published somewhere. Yeah, that would give us good data to maybe work further on identifying that. Genetic tests to identify those animals which are indeed efficient in producing milk. And this is the other one, a complete mess. Data is scattered all over different systems, different data dimensions. We have a lot of issues with people that are, let's say, are trying to work together but having difficulties because they don't speak the same language. In Europe, that's sometimes very difficult to have those big projects, but then people very subtle, they don't understand the language very well, and then because of that, we are not fully accelerating on really the data-driven approach that we should have. Then the validity of the data. Yeah, this is a Dutch calf that I use as a joke. You see probably the unique identifier. Yeah, so one ear tag is 5 at the end, the other one is 6. This is happening. Yeah, so you need to check the data. We did some artificial intelligence random forest predictions some years ago with a PhD student, trying to see if we can use, for example, DERICOM 305 or PC Dart data to use that data in the field, or sorry, later on, not in the field. So can we use that field data later on, or do we first need to clean it? And you definitely need to clean it. We very rarely see people being honest as scientists about that data quality. We know, yeah, that in animal science, if you're doing animal science, yeah, we know that there are issues, and maybe it's those animals that that have issues which are very interesting. Yeah, don't throw them away from your data. Maybe it's those animals that became sick and you wanted to exclude it in the trial. Maybe that's the ones that I want to know about. Yeah, be honest about what you report and especially what you not report. That's something that researchers should do. Then the other thing is very simple. Yeah, we see, for example, these are now 3 publications done about lameness detection. Lameness is still a major problem in dairy cows. We have automated detection algorithms that are published, but they're not implemented. Why is that? We still have that issue. We still have this farmer which has 3 screens with different data sources, and he's very, let's say, reluctant to have them talk to each other because it could mess up the whole thing. Is our industry really willing to share the data? Do farmers get any benefits? I think at this moment, yeah, it's a value ever flowing back to the farmer. We all say that the industry, in our proposals, that the industry will benefit, but do we? Yeah, do we have profound knowledge of integrating all that data together? Yeah, this is, I think, something that we need to address more. We need to become fair. Fair is something which has been right now quite active in my part of the world. Fair principles. Make sure that the data is findable, accessible, interoperable, and reusable. Yeah, we should try to produce that kind of data, and that's not easy. I now have a Horizon 2020 in which this is going to be a very big component. I have another one together with human colleagues where we're going to create a network to see that we always talk about One Health, but let's make it real. Let's try combining data sets from the human and the animal part. Yeah, and that's when we can be fair. I've been doing this quite myself. Yeah, practice what you preach. Reusing existing data. This was a data set that we published now already 10 years ago about milk production data on a transition disease data set. Now we're reusing that continuously. Yeah, and even now we started in publications, yeah, about milk bot lactation curves, but we reuse the data, yeah, trying to see if we can go further. Yeah, existing data models. We even now did a GWAS study on top of that lactation curve milk bot modeling. Yeah, and try to build on what others have done. And this is something that people often overestimate or don't do enough. Reuse what's already been done. Reuse data. There's nothing wrong with republishing what has been done, maybe in another way. Yeah, because that's, I hate the word innovative. This paper was innovative. Yeah, that means that it was the first. What I want multiple papers that reconfirm what others have found as well. That is something that we don't do enough. I want to also show 2 short use cases on what we have been doing, illustrating, again, practice what you preach. The G+E project was a large genotype by environment project in which we were combining data from herds and then come up with, let's say, management tools for genomic selection. In short, what we have done was a multi-research site study where we had extremely horizontally partitioned data, a lot of different industry partners, where we combined data from these research stations, and then first we brought them together into, let's say, it's kind of a standardized data set and worked from that. And our question was, can we create some novel indicators of the metabolic imbalance during transition and their association with production parameters across these research farms? Can we find something which is measurable, an indicator for the transition period? So we had all these different stations which had then, let's say, their data manipulated into one data model, And then we created a very, in my opinion, of course, I can say that, nice, let's say, data pipeline to find out indicators that would say this is a good cow, this is a bad cow in her transition period. So this was eventually done in a pipeline in which we tried to find out these animals, but also we want to be, because these are research data, and this is not practical. In practice, we will never be able to do this. So what we first did is on the research data, we tried to identify balanced animals versus the others. What was the difference in those research stations? Well, the balanced ones, these were animals that were producing the same amount of milk as their herd mates, but they were eating 2 kilograms more in the beginning of lactation, and because of that, their metabolism was saying, this is okay. I'm not overstressing my metabolism, my liver metabolism, my fat metabolism. I'm doing this in a more easy way that, compared to the others. And especially the dry matter intake was different in these animals. But of course, as I said, this is research data. So we also came up with the question about, can we predict this? Can we look at biomarkers in milk and find out if we can predict this animal that's good or bad? We used different methods used in Europe, and eventually used also the mid-infrared. So you use a mid-infrared spectrum in the milk to see if you can identify those animals. And then, of course, it's maybe even possible to do normal milk control and identify the balanced animals, and then move even further and do a GWAS on top of that. So we did the test from the 3 milk biomarkers. Eventually, we choose the best model, which was the mid-infrared, and from that we did the mini GWAS, and we did the larger GWAS together with CRV, and we were even able, with that prediction using MIR, we went on and said, let's look now at the GWAS component in that. Is being balanced heritable? And yes, this was recently published with my co-authors. We've identified regions highly associated with the metabolic clusters. So we know that there's a component, and this is similar to heritabilities, which has been published for other metabolic, let's say, indicators. We can identify, reusing all that data and creating a very nice pipeline. We can create even a larger prediction GWAS then, which confirms those metabolic clusters. We learned a lot about this. This was not an easy task. Multiple mistakes that we made. It took us 5 years to develop this, and if you think about that, that should be quicker. I'm sure that we can do this in a better way. But let's say overall, I'm quite happy with the results that we had. I want to show you one last thing, which is now actively on my scope, and that's all the screens. Let's think about that. We had the idea I had a guy from the PhD student from the economical department, and he came to me with the fact that he loved deep learning. Deep learning is a technology which Google is using, for example, for the image analysis. I think this will also be part of the next presentation. And we had the idea, can we represent, just like Google is representing us and learning from us, can we represent all the information that is in all the different systems on a farm? Can we represent this in a tensor? And a tensor is a kind of an algebraic object that describes the multilinear relationship between different, let's say, objects in a vector space. What we had the idea about is, can we use all this information to create on top of that a TensorFlow, which is a deep learning algorithm, to represent lactation curves again. So what we did is we very simply looked at milk production lactation curves, because we wanted to kind of find a way for algorithms to look at milk production in the current lactation. But our main goal was, if we can do that, and this is the deep neural network that was published very let's say last year, about using all the information on a farm to represent, let's say, lactation curves right now. But what we wanted to do is we wanted to do this first to try the next, because if we have this neural network, it would be awesome if we could then next create the subsequent lactation curve. Because what we can do then, is we can say, look, farmer, if you combine all the information from your farm in that tensor, you can try to predict the milk in the beginning of lactation when the calf has freshly calved. And we were able first to demonstrate that we can do this very accurately in the current lactation. And you can see these graphs that illustrate that. So we can look at the lactation right now, and then we can fulfill the remainder. That's something that has been done with Woods curve milk models, nothing special. But what we were not able, and this is something that I will skip in order of time, what we were I think one of the first to publish this is that we were able to create a deep learning network that is predicting the subsequent lactation. So given everything that you know from the farm and the cow, what is going to be her lactation, the next one? Because you just add in the same tensor, you add, let's say, the information from the previous work, yeah, and then ask the network, predict now what's going to happen next. And now it says Liseune et al. In review, but this was already accepted as well. So let's say the neural network was created a little bit more in a more complex way, but very interesting. Now we can predict on a quite accurate way the early milk yield that cows are following, given everything what you know up to that moment. And our model outperformed any other published literature, but the published literature is mainly using the milk production from the current lactation, but that's not what you don't know. Yeah, if a cow has calved, you want to have an estimation about how good is she going to be. Maybe you say, yeah, maybe that's her herd mate, or it's the herd average. Or so we use different, let's say, methods to compare, and found out eventually that our subsequent lactation milk yield predictor works very well. This is now published, and especially what we're wanting to do is we want to see if we can use those predictions to see an animal that has performed less than what you expect. Yeah, of course, that's interesting. Maybe we can detect early disease by doing that kind of predictions, or use it as a herd monitor. So, and with that, we're now looking at let's say, making this more complex in a federated way, but I will keep it with that, because otherwise we'll keep talking, because I see that I think I'm already a little bit over time. This is my son. I prime him to become very nerdish as well in the data component as well as the dairy component, as you can see. And with that, I will leave the floor for some questions if possible.
Okay, thank you, Miel. Excellent. Yeah, we are running over time, so there is a question in the chat, but maybe you can answer that in the chat. But I think, yeah, it was very good to have you talk about well, the applications on the one side, but also the challenges, right? Because that's really what this AG2PI project is about, to identifying what are the challenges moving forward. And I think the V's that you indicated are very, very good way to characterize that. So I think, unfortunately, we have to move on. So, but if you can go to the chat and maybe answer that question in the chat. And so our last speaker is Dr. João Dorea. He's an assistant professor in the Department of Animal and Dairy Sciences at the University of Wisconsin, and has a joint appointment with the Department of Biological Systems Engineering. And so, you know, this transdisciplinary research, I think João is a good example of where we need to move to. And this focuses on precision agriculture and data analytics, and specifically dairy cattle. And he's going to talk more specifically about image analysis related to dairy cattle. So João, the floor is yours. Are you able?
Okay, can you guys hear me and see the screen? Let's see.
Yes, it's not in presentation mode yet. And now we see the presenter mode, but not the, here we go. Yeah.
Okay. Alrighty, thank you very much, Jack and Agriculture, the AG2PI for the invitation. And it's a pleasure to be here and talk a little bit about the aspects of computer vision related to challenges and opportunities that we are facing in the dairy cattle side. And thank you for trusting non-geneticists for talking about high throughput phenotyping. So I will start actually with a slide that I got from my colleagues, the geneticists in the department. So if I'm saying something wrong here, I'm gonna blame on them. But the importance of the high throughput phenotyping and what is associated with this, it's very clear in the genetic side, which animal geneticists and breeders, they are very interested on understanding how the DNA information moves through intermediate phenotypes and endpoint phenotypes. And the omics technology is really helping the studies and the research in this area, especially with the price and the technology used in this for DNA, for the omics in general. And it seems that the bottleneck, it's in the endpoint phenotype. So thinking about these, specifically thinking about complex phenotypes or novel phenotypes that are really hard to measure, these would be a common place, a common interest, not only for geneticists, but also for people working in the precision livestock management that are interested in this for farm management decisions. So one thing that we're gonna be discussing here, it's how we use technology to get these phenotypes that is from common interest from geneticists and from people working in precision livestock farming, and what are the challenges related to that.
So Zhao, you want to move the mic a little bit closer to you?
Yes. Maybe I'm moving the mic a lot. How is that now, Jack? Is that better?
Good.
Okay. So in this context, when we think about agriculture systems, right, and the importance of these.
Actually, it's worse now. Sorry.
Okay, maybe I'm gonna pick the mic up. Or is now it's better?
Yes.
Here. Okay. So when we think about the high throughput phenotyping environment in this space that people work in, we can clearly see that in agriculture, it's much more advanced, right? So the data integration initiatives, the people research working on the crop side, they are ahead when you compare this with livestock. And the integration of weather, crop, soil, and water in the agriculture is more advanced compared to livestock as well. Not only the high throughput phenotyping related to specifically the plants or crops and animals. Right? When we think about livestock system, the data integration, the initiatives in the data integration side is still in early development. It's still really difficult to collect animal phenotyping. So the animal level information is still a big constraint when you think about data integration. And this is extremely important when you think about farm management decision and genetic selection. The phenotype is a big component of that thing. And because of this, we are interested in sensor technology as a way to generate those phenotypes and specifically generate phenotypes that are associated with the animal level phenotype, let's say, put this way. So although we have data from weather, crop, we have some animal phenotype technology, animal phenotype information as milk yield and milk components, we still miss a lot of important phenotypes like animal behavior, body weight, body condition, or and that's why we are very interested in using the sensing technology, and we have different options that are sometimes associated with specific phenotypes, and sometimes could be shared to collect this phenotype. And today we will be talking more about cameras and computer vision systems, although we have worked with wearable and infrared spectrometry, and Mills just show applications using other type of sensor technology. So today we talk more about the computer vision.
Zhao, you're still fading in and out.
Okay. See if I'm gonna get close here and try to speak loud. So let me know if you get better, and if you get worse, let me know again. Okay.
Okay.
So so thinking about the computer vision, why we be talking about computer vision as a technology for high throughput phenotyping is that the image can generate a lot of information that is extremely useful and helpful for usage that goes beyond the application that someone developed. So in here, just put an example. You have in an image the possibility of collecting animal phenotyping, for example, behavior, right? So you can predict if the animal is standing, lying, and things related to behavior. But also in the future, if you want to come back to that source of data and that information related to weather or related to the surrounding environment, housing system, and social interaction, you can come back and do this. While if you're using a specific wearable sensor, for example, and you do not have label for that, so then you're probably going to be restricted to that specific application. So the power that we find in images and in computer vision system, it really are huge and can expand and can be used in the future applications. How is the audio, Jack? Is that good?
You're still fading in and out.
João, don't move. Keep looking at the computer. It seems that when you're looking forward, it's okay.
Okay. Okay, let's do this. Okay, so when you think about those computer vision systems, we have different type of data, different source of data. Just to put in a context, we can have RGB data, which are the images that are regular collected in your cell phone, right? This color image. We can use thermal image generated from infrared thermal cameras that can give you temperature of objects in the scene. We can have depth image that can generate a 3D representation of the object as well. So a computer vision, some cameras that can generate most of those images at once can give you capabilities of studying and generating different phenotypes as you decide to do this very and with the interest in computer vision systems, because images can generate more phenotypes in one single data collection, for example, the interest in how we analyze that image and the interest in the deep learning and machine learning algorithms as we extract the information on those images are also growing a lot, right? And the framework that is being already developed by other industry, it can be a powerful resource already developed by someone for us to use in the event. So in a recent review, we select papers in animal science that try to predict an animal phenotype, and then using deep learning only, right, to generate this phenotype. And we select 44 published peer-reviewed articles. And what's interesting in that review was that 84% of the articles, they were published in 2019 and 2020. So it shows very recent interest in the use of deep learning for computer vision, which is already proved as an excellent tool. But in our field, it's very recent work, and it's still not a lot of work, but considering for 2 years was a lot of published papers. And when you look at the type of image that people are using on those applications of animal science, computer vision, the majority is RGB, the RGB image, and the type of tasks that are performed to generate those phenotypes are image classification, which could be considered a more basic task that basically they're going to detect things are in the image or not. We need to consider that some of the deep learning for image classification are used for feature extraction, and that's why it appears as half of the total papers using image classification as the main task to generate an animal phenotype. When we think about using deep learning and developing computer vision systems, we face problems as the same in agriculture and crops. In livestock, we face the same problem, and these problems are related to high dimensional data sets that are collected. And that problem, to be more specific, those dimensional data sets implies in large data sets, heterogeneous data sets. We collect a lot of information. Sometimes we need to merge that information with the data set, that tabular data, and then it's not a trivial task to be performed. And most of the time, those images, they do not have labels. So we have cows there, but we do not know where the cows are, which cow is which. So train the algorithm that we use. And I would say that the majority, it's a supervised learning. So we need labels. We need to tell which cow is standing, which cow is lying. So we need labels for those images. And because the size is huge and we do not have label, putting labels on those images is not a easy task, or at least it's a very time-consuming and expensive task. So we have noise data and a huge redundancy in the data sets that we collect. So do we need to have images every second? Do we need to have image every hour? So that's something we're going to talk more specifically. And we also have special and time dependence in the data that is collected, and this also creates some difficulties for us when you think about large-scale data collection. So just to put in context about a little difference that we face, and this creates a challenge in livestock, that is related to longitudinal data, thinking about image. And in crop, when we sample and we take a picture of a plot, right, to measure growth or to measure health status of that plot or that plant, if it's a plant level, the sampling unit, it doesn't change in space. So you can come back every hour or every day to the same coordinate and get the information or take a picture of that specific plot, right? When we think about dairy cattle, in this case, I mean a dairy barn, and you are interested in feeding behavior, for example, your sampling unit is not fixed in space anymore. So the camera that is collecting that information is looking to a cow that could be in this position here, as you can see up in the slide, right? But if you train your model to recognize that cow in the feed bun using the first 3 cow examples here, and then you try to predict using another using image of a cow with a different pose, it's very likely that your algorithm will perform very poor. And we can see that in those 2 examples, right? So I can identify very well cows in the feed bun here, the first image. But then if I do not have a specific pose or view of that cow in my training set, I won't be able to recognize that animal because I do not have this example. So in this case, the cows are moving around and your object is moving, and the feature representation to identify these objects, classify a pose is important. What do we need to do in order to increase this representation and have a more robust and generalizable model make your inference? So what you could do is collect more data. That's the initial thought. That's what you do. Let's collect as much data as we can so we can create enough representation in our data sets to possibly cover all the animal pose and then identify the animal, or it can cover all behavior pose, and then we could cover standing time, or we could predict lying time independent of the position or how that presents in the image, right? But the question is, do we really need to collect more images just about collecting more data and try to cover as much as we can? So when we started doing this, we started also creating a problem. We started creating a huge data redundancy because the data that is sampled, for example, every second or every 5 seconds, in most of the cases will look the same, right? So we have here, for example, 2 hours of data, 1,440 images, and it's pretty much the same image. We have one calf changing a little bit the pose, but we have pretty much the same image. Okay, so do we need to collect all that information? Do we need to store all that information? Do we need to train a deep learning or machine learning or any model using everything, all the data? So that's something you need to think about, because when you look all the time stamps in the data set, we can see a much better feature representation. So animals are moving around. So if I identify the animal, I can collect different poses of those animals, and then we could possibly better train our algorithm, right? So here, just an exercise to show you the importance of that data representation, okay? So if we pick those 1,000 images that I just showed you before, and if you run a cluster and select 5 clusters, okay, for those images, we can see that we have, based on the cluster, different representations of those images. And these are related to the animal pose, as you can see here in those 5 clusters, the examples of the image that we have. So we have images that we have all the animals standing, we have images that we have all the animals lying down, and then we have images that are some animals standing, some animals lying down, okay? And we can see that some clusters, we have very few images, so it's very imbalanced. So if you train all together, we'll be missing more representation related to, let's say, all animals standing, we're missing information of the image in this type of representation, okay? So this is something that we need to keep in mind as we decide to collect more and more data, because what do we need to optimize data storage? We want to optimize data analysis. So collecting more data not only will result in better algorithms or better results. So if you do these, and if you try to optimize and have a better representation using the full data set, we can achieve very good accuracy to predict behavior. And I'm sure those behaviors would be extremely important in the management decision and also genetic group. So the opportunity of using the image analysis to predict behavior is huge. But one thing that we need to keep in mind is that we need to optimize resource as we move forward using those type of systems. Another thing that I want to touch here is the redundancy of the data is not only a problem for training the algorithm, but also for inference, okay? So do we need to make inference in using a trained algorithm every second? We need to run analysis in every frame. And what we have seen is that this is really phenotype dependent. We have some phenotypes that, yes, you need to run inference every second in order to detect an event. And some other phenotypes you can collect every hour or every day. So that's something that needs to be studied, and then you have a clear view for each phenotype. So here, for example, we have a drinking event. So the animal spent 12 seconds in a drinking event. So it starts in this image in 1 minute 50 seconds and finish at 1 minute 4 seconds. So if you're collecting images every minute, you're gonna miss that event. But if you look for lying time, so that's different. So the animal stay in this pose or lying for a longer time. So you should be able to sample in a longer interval the image and make the prediction. So that's something that you need to keep in mind as well. So just to illustrate, we are predicting the behavior of that animal in the feed bank, right? So every time that this animal is visiting the bank, I'm identifying the animal. Here's an easier test because the pose, it's always the same. So it's this, the animal position in the bank will give you the same pose, always different from the other application that I showed you. So the capability and the predictive result is pretty good to identify the animal every time it goes in the bank. Do we need to make inference every second? So when we compared making inference every second, every 2 seconds, every 5 seconds, until up to every 10 seconds, we didn't see any change pretty much in the total time that the animal spent the bank, okay? But these would have a huge impact in the amount of data that it should collect to perform inference. And when we think about that type of inference, so here, we are using the identification of the animal, tell if the animal is in the bunk, eating not, right? So the identification in computer vision systems should be our unique ID. So you want to be able to get an image, and this image generate the phenotype and attach that phenotype to a animal ID. The problem is that if you do not have coat color pattern as we have in Holstein, if you have only Jersey cows, how identify those animals? We use an image only. So we have tried to use the shape and different 3D representations to identify the animal. And those would include point cloud and voxels and others, right? So using voxels, we are achieving a very promising result to identify cows and calves based on shape only, based on not using color at all. So these would be extremely important when we think about integrating phenotypes as body condition score, body weight, right? And then you will need the ID of that cow that is passing through the lane every day, okay? So by connecting, again, the behavior and the animal ID, then we are able to have a system that is extremely powerful for farming and management decision and could give important phenotypes, hard to measure phenotypes for genetic selection and animal breeding programs. And we can reduce data dimension as well. So if we have, let's say that I do not have data redundancy, but now I have problem with data size. I have large data. How can I reduce the size of the data and still have good prediction, right? So we are using 3D images, as I mentioned earlier in the presentation, body weight, okay? And we are extracting features related to volume, area, length, and using those biological features that we know are related to body weight to predict body weight of those, okay? And we have a very good relationship with volume, area, all those image-based features with body weight, and that's why we can predict well body weight of those calves. We try different approach, but we can have a very good prediction. And we have few number of animals in this study, but in studies that we have published with larger data sets, beef and pigs, we have even better results when we use the 3D image to predict body weight. However, one question that came in our mind using this approach, which we segment and we can delete background information, we can only stay with the body of the animal and use this to predict. The question is, can we reduce this even more? So what we did here is we collect a vector in getting the maximum length of this depth image, and this will generate the representation that you can see here, this vector. And we collect in this yellow line here another vector getting the width of the animal. So we concatenate those two vectors and we create one single vector. So here's one side by side, two vectors, okay? And we got that vector. Now we have one-dimensional data, much smaller data compared to these or compared to the whole image. And we use a deep learning unsupervised method called autoencoders, which basically we try to map the input and reconstruct this input and generate the output, which should be similar to the input. And for that, we have a latent space which extracts features that are used to reconstruct the input. So those features are related to this vector, right? So our question was, can those features be associated with the animal biometric and then body weight? So in this matrix here, you can see the relationship of width, height, and body weight, but also the relationship of the features, 20 features extracted using the autoencoder of one-dimension vector, this vector, right? The relationship of these with the biological data. So we have positive and negative associations with those features and the biological features. So what we tried was to use the features extracted from these one vector, one dimension data and predict the body weight. And when you compare with the biological data, we have very similar results. Okay, so a little worse for the computational, for the autoencoded features, but very, very close. But when we look to the reduction in size, we reduce from 600 kbytes to 0.21. So this is a huge reduction in data size, and these change a lot computational resource used and data storage. So that's something interesting from to reduce the dimension of the data. So things can get worse when you have multi-view system, much more cameras, cows move from one scene to another scene. There are different generators of data, but the cow remains the same. The cow changes pose, so these all create a complication. And when you scale up your system, this is one farm, this is one pen with 62 cows. Also, the size of the data increases a lot. And when we think about that, we usually treat that data set as a number of records, but we need also to think about size when we think about image, because sometimes the number of records for image doesn't mean a lot. So here we have 4 months of data and 40 cameras on the farm generating data every 5 seconds, every second, I'm sorry. And we generate 414 million images, or we consider that record. But these could be not big if you look at the number of records, but if you think about size, we're talking about 90 terabytes in 4 months. So size really it's important when you think about image maps. Right? So if you think about what an image is, one image is a 3 matrix all together, and that's why you scale that data in size more than any other tabular data or structured data. And it gets worse if you think about hyperspectral imaging. Hyperspectral imaging, you have even more channels, so the dimension of the data is much more, and the size of that really up. And that's why we're using cloud computer and distributed computer to train our algorithms for the data and to manage the data. So in our group, the guys working with us, I don't think they think big data is that really exciting. So that creates actually a lot of work for them to deal and to train the algorithm, complexity that it brings. So as a final consideration, I mean, the computer vision system, it's a very powerful tool for high-throughput phenotyping. It can be used for farm management and breeding programs for sure. It's getting cheap, precise. It can be implemented in real time, but we're still facing some challenge related to data dimension and processing. So integrated knowledge related to computer science, statistics, and the domain knowledge of using the computer vision is extremely important to advance the use of computer vision systems in livestock operations, either through farm management and breeding programs. Well, with that, I would like to acknowledge the group. I have a great group of guys working with us, and I have great collaborators at the department as well. And we have great support to run all those trials and make our program. So thank you very much. I appreciate the opportunity, and I will take in. Thank you.
Hey, thank you, João. There's a couple of questions in the chat. They're fairly specific. So because of the time, I'll just ask you to answer them in the chat or afterwards. We'll save these. But thank you very much. So because of the time, I'll wrap up this field day. I think we had another very good field day, lots of information, obviously lots of achievements from the first talk of what's been achieved in dairy cattle. In the last 2 talks pointed out lots of opportunities, but also lots of challenges on how to make use of all this data that's been generated. I think we had the maximum participants I saw was 160, which I think is very good. And there will be more that will be joining once it's posted, and the full recordings will be posted later this week, so you can always go back and share it with others that you think might be interested in this. Wrapping this up, we are looking for topics for additional field days. I think you now have a pretty good feel for what we're after in terms of these field days. So if you have topics you want to present or you know of others, please send them to us. Typically, field days will be every third Wednesday of the month, 10:30 to 12 US Central. Of course, we deviated from that this month, and we will also be deviating from that next month. So the February field day will actually be on February 24 because of another conflict. And it will be a field day that will focus on the seed grant program that we will be releasing early next month. So the seed grant RFP, and with proposals being due March 19. And in this February 24 conference, seed grant conference, we'll get into a lot of details of what we're looking at, and also try to find ways to connect people that might be interested in working on a proposal. These seed grants are not typical. We've talked about this before, and actually a lot of the points that were made by the three speakers in terms of the challenges of the V's that Miel talked about, I think are excellent opportunities for putting a proposal together on how to start addressing these challenges. And that's really what the seed grants are about, is addressing challenges that are emerging as we get into this big data and using it for genome to phenome research, as well as, convincing society that this is a good idea. And so more details on that in the RFP and on February 24. And then also we'll have a training workshop. There was one earlier this month on data science for biologists, and in March, the date hasn't quite been set yet, there will be a training workshop, the reverse, so genetics for computational scientists. So be on the lookout for that. So with that, I want to thank our three speakers again very much for their presentations and the time. And I think, like I said, it was very excellent. And thanks for all of you for joining, and we'll hope to see you next time. Thank you.